So in this video, we're going to be going over protein structure. Protein structures is crucial because its specific 3D shape dictates its function, which enables it to interact with other molecules, catalyze reactions, provide cell structure, and transport substances. Even tiny changes in shape, like a single amino acid swap, can cause severe, issues. An example of this is sickle cell anemia, which is caused by an amino acid swap. This highlights the importance of its structure and its function in biology and medicine. Protein structure can be divided into 4 levels: the primary, the secondary, the tertiary, and the quaternary. The primary structure is just the linear sequence of amino acids determined by peptide bonds, as seen here. Right? And for a secondary structure, it describes how the backbone folds locally, which is mostly stabilized by hydrogen bonds. The two motifs that make up this secondary structure are the alpha helix, which is a coil stabilized by hydrogen bonds running out along the helix, and the beta sheet right here is like a zigzag type of pattern stabilized by hydrogen bonds between the strands. Right, so here you have the beta sheet, and then you have the alpha helix right here, describing the secondary structure for proteins. Now next, let's talk about the tertiary and quaternary structures. I know this looks like a lot, but I'm going to break it down one by one. So tertiary structure is just the higher order folding within one polypeptide. So it's just describing how distant parts interact. It's stabilized by hydrogen bonding, van der Waals interactions, hydrophobic packing, and sometimes disulfide bridges as well as ionic bonds. And so here, if you look at the diagram, you could see all these different types of bonding mechanisms being active in holding this structure together. So in an aqueous environment, polar groups tend to face outwards, interacting with water, while non-polar groups pack inward, and this is called hydrophobic packing. So this is depicted by the hydrophobic interactions, which is seen on the inside of the protein, away from the aqueous outside environment. So disulfide bridges form when cysteine side chains oxidize and form a covalent bond between sulfur atoms. And disulfide are favored outside the cell because that environment is more oxidizing. Inside the cell is more reducing due to antioxidants. And for the quaternary structure, this describes interactions between multiple polypeptides. Each polypeptide is a subunit. 2 subunits is a dimer, 3 is a trimer, 4 is a tetramer, and so on. That's going to be it for this video. Thank you for tuning in, and I hope you learned something.